In the field of testing methods for viruses, there has never been greater progress than in recent years, due to the corona pandemic. Now, researchers from physics, nanomedicine, and virology have developed a measurement method that could overtake previous methods in a hurry. COMPASS is an extremely fast testing method, and the actual measurement takes just 10 milliseconds, the blink of an eye, and the whole procedure of putting the rod into the nose, into the buffer, and into the device is over in less than 10 seconds. And that really distinguishes this new measuring method from PCR, ELISA, and the like, which are extremely time-consuming. The measurement results can be read directly on the battery-powered COMPASS device or via Bluetooth on a mobile device such as smartphone or laptop. With COMPASS, we can detect the effective viscosity of magnetic nanoparticles, which means that we have nanoparticles that are specifically functionalized and the appropriate counterparts that can then adhere to the surface. And now you can imagine that nanoparticles have a certain size in terms of diameter, and if something adheres to it, it gets bigger. What we do now with external magnetic field is we wiggle these nanoparticles, we rotate them, so to speak. Now we know this as with figure skaters. With their arms outstretched, they rotate slower than when their arms are on their body. And that's exactly what we see. In other words, we can measure these differences in speed and thus infer the size of these particles. Or to put it another way, we can see whether something has attached itself to these particles or not. COMPASS is now our special approach to be able to precisely measure these size changes extremely sensitively. An important piece of the puzzle for the development of the measurement method was contributed by the working group from the Section for Experimental Oncology and Nanomedicine at the University Hospital in Erlangen. The team specializes in the functionalization of magnetic particles. These particles are iron oxide nanoparticles that gives us the magnetic signal. Due to the special measurement methods that we use here, our iron oxide nanoparticles are supermagnetic, and therefore we have the ideal prerequisites for this measurement, so that we get a very good magnetic signal that we need for this measurement. For functionalization, we need particles that have a surface, a coating that enables us to chemically bind them so that we can bind our target via linker molecules. For example, an antibody or an antigen, which in turn enables us to bind the counterparts with the particles as a whole in the device and then generate a signal by changing the mobility of the particles, which we can then measure. To be able to determine the coronavirus using the test method, the physicists needed sufficient samples from patients with different viral loads. The Institute of Virology and Immunobiology was able to support them in this. The physicians there also recognize the advantages over other test methods. The advantage of the COMPASS procedure is, of course, that we can do the confirmation test in one step, so to speak. COMPASS is very suitable for use at certain locations where you want to test a lot of people in a short time, such as theaters, cinemas, concerts, or something like that. You can maybe also imagine it in the pharmacy. Alternatively, COMPASS could be used as a bedside device, so directly on the patient, or even in the emergency room as a point-of-care test. If you look at it from an economic point of view, this measurement procedure with beads provides a different basis than the previous procedures. And these beads can be produced simply in large quantities, which are not as costly as producing a PCR test, for example. And that is why it is so much more cost-effective, and the costs for the devices are also lower, simply because it can be produced using a 3D printer, for example. And of course, there is also sustainable potential for the future.